is a research institution based in Palau, and our main objective is to provide scientific data for management of our natural resources. And so far, we have been able to get data from the surface for our pelagic ocean here in Palau. And this partnership with OET is a really wonderful opportunity to get deep sea samples as well for eDNA, which is environmental DNA. So for those listening.
Oh, you can move it. You can move it anywhere.
It's a really cool critter on screen right now.
This is a crinoid. It is a type of echinoderm, so related to things like sea stars, sea cucumbers, sea urchins. The crinoids are one of the older echinoderms that we often find in the fossil record.
Do you think we have time to do a snap zoom into that little thing in the crack?
Yeah, there's a little anthomastus, I believe, in
I'm
there.
always a fan of a snap zoom.
You can push in there.
Maybe try to take...
So we have a small anemone and the small mushroom coral.
Nice. Great.
Which one is the mushroom coral?
The mushroom coral is the pink one. It has the 8 tentacles with the little pinnate feather-like structures
The one
along
in
those
focus.
tentacles. That's how you know it is an octocoral.
Octocoral.
So it's a type of soft coral in the subfamily Anthomastinae. This is an anthomastus mushroom coral.
All right, come wide.
Yep.
All right. So yeah, as we head up slope, we're gonna when we arrive at 1,000 meters, we'll plan to take a Niskin, which will be part of our eDNA sampling program. If you have anything else to share, feel free, Elsa.
Thank you. Yeah. So as I was saying, we would be taking the water samples using a Niskin bottle at the predetermined depths intervals, and the next one would be 1,000 meters along the slope.
Yeah. And on this dive, since we started the dive at around 1,560 meters, we have one extra bottle open, so if we end up seeing some high biodiversity spot or want to trigger 2 Niskins at a particular location, that's something that we can kind of decide on the fly.
May, who's in charge of taking still camera photos during the dive?
Yeah, so Elsa is working on it, but
okay.
We're having some issues with the still camera.
Yeah, Roger.
I don't know if you got the memo, but yeah.
I heard some stuff, yeah. I was just curious.
Yeah, something a little bit tricky with the focusing, and there seems to be some kind of almost like smear or something like that.
Yeah, it was packed away for a year, so it could have collected some dust and muck on it.
Yeah, it's definitely not looking like how it used to, the outcome. So we're doing our best, but even when we're completely still,
Snap
it's...
on the star.
Yeah.
Yeah.
Get a snap on the star. Snap scene.
This is a Benthopectinidae sea star, likely benthopectin.
Nice. All right, come back wide.
Thanks.
So as we make our way upslope, we're kind of going to start getting into a little bit steeper terrain soon. And in terms of the kind of samples we're looking for for the rest of the dive, we've got an entirely open set of slurp chambers, but all of our other boxes have corals and rocks or a squat lobster that we really don't want to have escape. So
Oh yeah, those are fast.
yeah, and it might be a new species actually. So
Oh, wow.
we're a little bit off limits on our bio boxes. So we're gonna be kind of keeping our eyes out for
Sample jars, small
yeah,
samples.
exactly. Slurps and niskins. And in terms of slurps, we don't have any fish on this dive yet.
Okay.
So if we happen to see an interesting fish and Jeff chimes in on the chat, or we decide that it's a good call, then that would be a good candidate. But otherwise, we'll be just doing some good zooms and getting some nice video to characterize.
Sounds good.
Yeah, we probably won't see very many fish until we get closer to the top of this feature.
Exactly. That's why we kind of want to make some headway. So that's the goal.
What's the depth at the top of the feature again?
690 meters.
690, okay.
Yeah.
Is that the shallowest we've gone in Palau so far on a dive, other than
Yeah,
ascending
that will
and
be our
descending?
shallowest. The last cruise, I think our shallowest was 750.
Nice.
Yeah, this morning we were really cutting it close, finishing the mapping on this feature. We missed the top just a little bit because it's so tall that we had to go back and map right across the top of it. We finished mapping at like exactly 3:30 and stopped and got ready to dive.
Go mapping team. Well done.
Yeah, Rennie was furiously producing all the products that we need for our dive plan.
Yeah, and so if you're wondering what that means by missing the top, so when we map, it's a function of depth.
Push in here.
If we miscalculate the kind of depth that we're looking at, then the line that we're going along with the ship, the sonar that reaches the seafloor might not actually cover that depth. So the shallower you get, the narrower the swath of sonar is. So that's what Megan was just talking about.
Yeah,
This is really really interesting.
This is a kind of third urchin. So one of the urchins that are called the pancake urchins.
Ooh,
Yeah. They call them that because when you collect them, their test is very thin, and so when you get them on the surface, they kind of pancake. That name came about when animals used to be dredged up from the seafloor and then identified, and so it hadn't been seen in its natural habitat until, more recently. And it took us a while to realize that's exactly what this animal was, because they're not very pancakey on the seafloor. They're more cushiony.
well, I would argue that pancake batfish are also not very pancakey.
Yeah, I think some common names can be a little misleading.
Yeah,
You mean like when they have eel in their name and they're not an eel?
Yeah, and then they're actually a fish.
That's a bit misleading.
exactly.
Yeah, all the things called lobster that aren't lobsters.
And all the things that are called cookies that are
Not
echinoderms.
Not actually
not
edible.
yeah.
Or raviolis. Yeah. You can tell scientists are
We're always hungry.
hungry on watch and falling back on things that they know instead of inventing completely new words.
I helped collect a sponge in the Clarion-Clipperton zone that we called the onion ring sponge because it was super paper thin, and it was the perfect ring. It looked like an onion ring.
It's funny.
Were you hungry?
I was definitely hungry.
Some pretty interesting rock formations.
Yeah, so this is actually carbonate, because you can see how it's kind of eroded away.
Yeah.
It's interesting.
Yeah, it's really neat as we get closer to the top.
It looks like art.
It does. It's modern art on the seafloor.
Sorry, May, were you saying when we get closer to the top, we'll see more?
More carbonate, yeah, definitely. So carbonate rock is formed from the remains of old coral reefs. And as we are exploring the tops of these seamounts, that's the kind of rock formations that we're going to be seeing covered in a lot of sediment. And all of those, that dark black coating that you see on it is manganese oxidation, and the kind of texture that you see is a mineral habit called botryoidal texture. And when we do these snap zooms, you'll be able to see that texture a little bit more closely, and it looks like a lot of little balls, and that's where the name comes from. Botryoidal actually comes from Greek, and it means grapes. So another food reference, but it's a very grapey texture, essentially, is what that means. And that's how that manganese oxidation is known to, that's how its shape is known to form on top of pretty much any sort of substrate that remains in the deep sea long enough and is interacting with the seawater and oxidizing. There's a fish, yeah. Let's try to look at that if we can.
It's a halosaur, so I think it's probably gone.
Yeah, they're pretty swift.
Look
And
at
a little
that
jelly. Nice. In our deeper depths, this region is volcanic, and we'll see volcanic rocks, so basaltic blocks or
Not a chance.
exposed volcanic rocks and rubble fields, things like that, but those will be at deeper depths.
We can take a look at that chrysogorgid up in the upper right.
Yep.
This area looks like a labyrinth.
It definitely does.
Push in there.
Nice.
So this coral is a Chrysogorgia, and then in its branches you can see a squat lobster, and it's a Europtychus squat lobster. We often see this association between Chrysogorgia and Europtychus.
So are they eating the coral, or?
No, they just live in the coral, and they help the coral stay clean, and the coral provides a nice habitat for the little squat lobster to hang out. So it's protected and it's up in the water column where it can feed more easily.
Okay, come wide.
Got one of those shrimp. Some brittle stars also around.
And the Nematocarcinus long-legged shrimps are very common in these types of areas. We see them all over seamounts, really.
Yeah. This is really neat.
And with how conical this feature is, I was really surprised to see the carbonate starting so shallow, or, well, it's a little deep. I mean, I don't know, maybe it's about average for this area. But I don't know, I think I expected it to
Where's the
be
fish?
almost all basalt. Oh yeah, there's a little cuskeel.
Oh, nice. Good eye. We could get a zoom on that. That would be great.
I'm thinking monometopus.
You can push in there.
Yeah, cuskills really like these little overhang areas.
Nice. Oh, it's got a lot of good hiding
Yeah,
spots.
there's so many like little tunnels.
Yeah, it's a great spot. The little shrimp inside. All right, I think we're good.
Okay.
Yeah, we could go check out that little cookie star too.
Yeah, the white one off to the starboard side.
Mm-hmm.
Sounds good.
All right, push in there.
So this looks like Ceramaster. So this is a sea star in the family Goniasteridae, also known as the cookie stars. But I wouldn't recommend eating it. That's great, thank you.
Nice.
Okay, come back out wide.
Oh, there's a cool anemone.
Hmm.
This
Take
is,
a look at it.
yeah, we
Yeah.
can look at it. This is Actinurnus nobilis. I don't know if it has a common name,
Push in there.
but maybe we can come up with one. It's very distinctive with its sort of white and purple coloration and folded disc.
It's really beautiful.
Isn't it?
Yeah.
Yeah, I like this one. That's great, thank you.
Okay. Come back out wide. Another one up to the left.
Can we check out that black coral to the right? Just a quick snap zoom would be great.
All right, go for a quick snap zoom there.
Looks like a Stauropathes. All right, thank you. That's
Okay.
perfect.
Back out wide.
Nice.
So with the sample that they collected earlier on this dive, they would be able to tell how old these rocks formed?
Say that again?
Like with the sample of the rock they took from the earlier,
Yeah.
would they be able to tell how old?
Oh, how old they are? Yeah, exactly. That's the goal. So, yeah, we're trying to collect some volcanic rocks from throughout the Palauan waters on these two expeditions to be able to age date some of the features that we're looking at, these seamounts and ridges and things like that. So,
Push in there.
Fishing.
This is a snailfish, maybe? No. It's a cuskeel. It's very small.
very small. Yeah. Often when we've collected fish, I've thought of them as being bigger, and then they show up in the lab and they're so tiny.
Oh, yeah, yeah, those two little... Oh, look how beautiful.
Oh, yeah. Incredible.
Coming in for his close-up.
Smile.
Oh.
Nice.
It's too close for focus.
Very
Apologies.
curious.
I'll zoom out.
Yep.
Jeff was happy with that. He said it's not a Basocetus.
No.
It
Nice.
had quite beautiful detail on its, was that its lower fins, Megan? Or May?
Oh, yeah, there was, there's some fin rays that were really nicely splayed.
It's whiskers.
We have a viewer that says the carbonate is a very karst shape. What is karst? Sharp?
Yeah, I guess the viewer is referring to kind of the way that the carbonate is eroding away. We see these kind of like pockets and pillars, and it's not a completely flat sheet.
There's another fish coming up. I can see it in upper.
Oh, yeah.
Wow.
So this fish is Aldrovandia, a type of halosaur.
Push in there.
I thought it was upside down for a second.
No, that's just how they swim. They hang out that way. Oh, yep, and then they swim away. They're very fast. So does anyone in the back row know anything about Palau's geologic history?
Could you say that again?
Palau's geologic history, like how the islands were formed?
Yeah, so Palau is a geologically very rich area that has a lot of history with volcanic activity, the other kinds of rock formations we see. There's a lot of limestone and carbonate, and kind of erosion is at play at the moment. So the islands were formed around around 40 million years ago. The Palau volcanic arc remained active for about 20 million, until about 20 million years ago. And the northern islands are more exposed volcanic rock, while the rock islands, which are maybe what most people are more familiar with in terms of Palau's sort of really beautiful features, these sort of floating like islands. These are raised coralline reef structures. They're Palau's limestone. So those were formed several million years ago, and basically as they erode away, they kind of crumble back into the sea, and erosion caused these sort of islands to create these discrete emergent remnants of former coral reefs. And what we're diving on, though, is much, much deeper than what most folks are familiar with diving in the rock islands. So the seamounts and ridges around the Palau National Marine Sanctuary, or in the Palauan waters, which is where we are right now. We're just outside the National Marine Sanctuary, but still within Palauan waters. Those are a function of volcanic activity, and we're here to characterize this geologic setting in the deep. So we have a general idea of how these seamounts formed. As Palau is kind of situated on the edge of the Philippine plate, and is currently part of the Palau-Kyushu Ridge formation, which is an old volcanic arc. So subduction isn't really active, or just very, very slowly moving along the Yap Trench, but it's more active in the Mariana Trench. So it's a little bit of a complex geologic setting, but it's a back arc setting, and we're here to collect these geologic samples in order to actually date some of the volcanic features. And as we get shallower, as we are now, looking at these old carbonate shelves and really interesting eroded carbonate features, this is where we're also able to see a lot of these really ancient communities of life. So corals and sponges and all sorts of other deep sea organisms.
Little tiny fish.
Yep, gopher fish.
Yeah, thank you. That was very informative.
Yeah,
Push in there.
Palau is really cool, geologically speaking, and as well as its sort of general location within a lot of different currents. So the current regime in this area is really important in terms of the nutrient availability for life, both in the shallow waters, and we also believe in these deep waters. So on this expedition, thanks for the zoom. Yeah, really nice. Yeah, so on this expedition, we're kind of planning our dives in parts of Palauan waters that really hasn't been visited with deep water capabilities. So beyond scuba depths and previous work that has been done in this region has been done at much deeper depths, more trench depths. So we're kind of hitting that sweet spot where we might see something really really new, and also be able to characterize parts of Palau's National Marine Sanctuary that hasn't been characterized before. So part of what the main goal is to look around, see what we see, collect the samples that we need, so small pieces of organisms, which we see quite a few of, in order to get that good habitat mapping references, so samples that will let us do further analyses in the future to learn about Palau's incredibly rich and biodiverse oceanic worlds from the shallow zones down to the deep.
Ooh, that was a cool Calyptrophora off to the left.
I missed it. Oh yeah, there it is.
Isn't it pretty?
Yeah. Yeah, we saw a couple of these at the very beginning of the dive as well. Very nice. Let's go take a look
They're just
at
like
it.
very satisfying looking.
Yeah, incredibly symmetrical.
Mm-hmm.
They look like a beautiful kind of candelabra. Is that a Paragorgia off to the
Hemicorallium.
Hemicorallian?
Yeah.
Nice. In the upper kind of bubble-like view, it's also quite cool to see.
Oh, sorry. I was looking at that sponge and then I hit my mic.
No worries.
There's one over here.
Yeah, that I think it's a Farreid.
The one underneath.
Yeah, or
Yeah.
maybe not. It's I definitely need to zoom on that one.
All right. All right, Aaron, go for a zoom.
What kind of sponge are you?
Oh, it's
I think it's a euretid.
Huh.
Kind of looking for the inside texture. Inside texture where the spicules are meshing together is telling me it's in the subfamily Corbitellinae in the family Euplectellidae. Yeah.
Very nice.
Yeah. So this is a softer, squishier sponge.
Very cool.
I was thinking it was a crunchy one, but this one's a soft one.
We have a crunchy one already.
Thanks. Yeah,
Okay.
I was like
Back what?
changing my mind at least 4 times on what I thought that was.
Soft or crunchy? Soft or crunchy?
That really is the question.
Hey Megan, so I had a question about the spicules that you mentioned. So you said that there are squishy and crunchy sponges, but they all would have the spicules.
Yeah, so they're all glass sponges, and and the spicules are made out of silica glass. But the way they knit together will affect the sponge's texture. So the euplectellids and the rosellids, they have more of like a mesh texture that can be more flexible. So when we collect it, it bends rather than crunches like a chip. Whereas the sponges in the families Euretidae and Farreidae have more of that crunchy, really sort of brittle, fragile texture, and that has to do with how the spicules mesh together to form that skeleton. The spicules come in many shapes and sizes. Some are very long and filamentous and easy to bend, sort of like a fiber optic wire, whereas others are very small and can be more like concrete-like, where they mesh together more closely, and then you'll have a more fragile texture. All the spicules in a sponge are unique, like the different kinds that are in a single sponge are unique to that sponge. You'll see similar spicules in different sponges. So like, for example, you'll have a certain type of spicule that you'll see within a family, but you won't see it in a different family. And when you get, you gotta look through all the different types of spicules, and that'll help you identify that specific sponge.
Thanks, yeah, really interesting. And the mesh versus, I guess, crystal, like a crystal-like structure makes a lot of sense in terms of squishy versus crunchy.
Yeah. Yeah, when we zoomed in on that sponge, I was looking down the osculum, that hole in the center, and you could see how it looked kind of fibrous, like the inside of a pumpkin a little bit. And that's the texture that I associate with the euplectellid sponges. Whereas when we look at a farreid or a euretid, it has more of a finer texture. And when we see one of those, we can zoom in, and you can see what I'm talking about. It's one of those things that's hard to describe in words. It's easier to see.
How far into the next waypoint?
We've got 130 meters.
And we're doing a Niskin at 1,000?
At 1,000, yeah.
Yeah.
What a beautiful piece of coral coming up.
Yeah. Let's definitely take a look at that.
So this is a hemicorallium, a coral in the family Coralliidae, also known as the pink corals, though this one isn't very pink.
Push
They
in on
can
the
come in many colors, shades of
polyps.
pink. The way to tell this coral from others is that the ends of the branches have paired polyps. Oh, there's a little hermit crab.
Little hermit.
There's some brittle stars. Looks like there's some zoanthids as well. Crinoids. So corals can be a habitat for many different species.
It's a really pretty shell with that hermit crab.
Mm-hmm. Yeah, picked a good one.
Yeah.
All right, come back, go ahead.
So something like that is probably like how old do you think? If you would give a rough estimate.
Rough estimate, that one is pretty large. I was looking, if you look at the thickness of the base, I would say maybe 200 years old. Could be a little older. I'm basing this off of growth rates for hemicorallium at shallower depths. The deeper you go, usually the slower these animals grow. So it could definitely be older than my estimate.
Thank you.
Also, you know, I'm measuring with my heart and my eyes instead of with the actual measuring tools and having the specimen in hand. Oh, there's some
That's a bit of
stoloniferous
purple.
octocoral right here on the edge. So stoloniferous octocorals don't create their own skeleton. They overgrow on other things. That can be rock faces like this, or old coral or sponge skeletons. This is Clavularia. You can see that the polyps are connected by a thin tissue encrusting the rock. And then behind it, you see that little thing sort of floating around in the current. That is a hydrozoan in the family Tubularidae. So it's a single polyp. I think they look really cool, like little flowers.
Yeah, a really beautiful shot.
Okay. All
Nice.
set.
All set,
Come
thank
on.
you.
So that's a really beautiful purple color. But in the deep sea, what could you maybe talk about why they would be that color? Is it a chemical reaction, or do we know that?
So in this case, the color doesn't harm or benefit the coral in any way. So yeah, the reason why it's purple is likely due to some other beneficial chemical that it has in its tissue that causes that color. Or it could be a carryover of color from when that coral might have lived shallower before it migrated down deep to live in deep depths. So we don't always know what causes color in the animals at these depths, but in general, the color isn't necessarily helpful or harmful to the animal.
Mm.
A lot of times we see animals that are red. Red can be helpful because that's the first wavelength of light that attenuates in water. And so if you are red, you're essentially black down here. At our depth of 1,200 meters, there's no light. We have to bring all of our own light. So we're perceiving the world very differently from how these animals perceive their world.
Thanks, Megan.
Ooh, bathy pathies. This is always a very satisfying black coral.
Found a nice little alcove
Yeah, it's like the perfect spot.
to get little whirls of currents.
I bet it like comes right in there
Exactly.
and starts to
That's what I was
mix
thinking.
around.
Push in there.
Especially since it's facing the rock face.
Exactly, yeah.
Oh, and
It's
it's
like
got 2 squat lobsters.
Nice.
You know, we often see black corals like this with associates. There's often a polychaete worm that might live up the center axis of the coral. Right here we see 2 Europtychus squat lobsters perching on the coral, as well as a little star.
Okay, come back wide.
Can we look at that shrimp sitting on the bottom? It looks like it has short legs.
Push in there.
Mm.
It's like
It's like,
slowly backing
no.
up.
It kind of looks like heterocarpus, but it's very skinny.
It's exactly what Jeff would have. So, same page.
You can see that really long serrated upturned rostrum. It's very typical of heterocarpus, but usually the heterocarpus we see are a deeper body. This one is very small. All right, cool, thank you.
All right, come back wide.
Nice. needed. Okay. Do we have any questions from our viewers?
That's what I want over there.
Yeah, so somebody's just saying shrimp, and somebody's saying it's waving high.
And it is waving high. Wow, look at this.
Oh, this is beautiful.
This is really, really
Wow.
big. Let's definitely take a moment to do some good zooms here.
So this is a madrepora. It's this scleractinian hard coral that's known to form reef-like structures in deep sea environments. They can form these large mounds and aggregations like this, and when they do that, you'll often see tons and tons of associates in the branches. So the more we zoom in and look, I think the more we'll see associated with this coral. Oh, there's a fish.
Push in there.
It's pretty impressive how it's kind of grown to take up the shape of the,
I'm going to zoom in a little bit.
yeah, the opening of the rock.
Yeah, it's like this is perfectly what I need to be shaped like.
Yeah, right. They fit like a puzzle piece.
Mm-hmm. Well, corals are known to have a very plastic morphology. They can change the way they look on a large scale to fit their environment.
So how old do you think this coral would be, Megan, if you had to give a guess?
I'm not really sure. I'm not familiar with the growth rates of this particular species, but it's got to be pretty old. I mean, it could be like 1,000 years old.
Nice.
It's just amazing.
Yeah.
Thousand years old.
That's my best guess.
It's amazing. Could just like stare at it all day.
Yeah, yeah. There's a lot in here. Lots of amphipods, anemones, brittle stars, squat lobsters.
Yeah. Interesting.
Yeah, I think if we could get like a pirouette around it, that'd be cool too.
Sure.
Yeah.
One Jake special, please.
All right, come on.
Originally, I thought those like little sort of polypy bumps were something other than the coral, but it looks like that's actually part of this coral. I'm not sure how or why that has formed that way.
That's kind of strange because it's not really consistent across the entire.
That's not really typical, but it could be that it was irritated at one point and like maybe it had something that caused that growth on it. I'm not sure. Oh, there's a crinoid and a sternostylus squat lobster. There are hydrozoans on this coral as well.
This is really, really fantastic.
piece growing on the bottom.
What's that?
I said it looks like a piece is growing from the bottom. I wonder if that's connected
Yeah.
or if it's separate.
Yeah.
Yeah, I think that lower piece down there is a separate coral, but likely related to this one. This one could actually be multiple corals too, that have grown together.
Like if it were to have broken and fell down, would it be able to regrow?
Yeah, like if we were to break off a branch, it would regrow from there. But if we knocked a branch off, it would fall on onto the ground, and unless there was really good current there, it probably wouldn't regrow. But that's not an impossible thing. I've definitely seen corals where they've toppled over, but the current in the area was enough to keep that coral alive, and it was able to continue to grow after the fact.
Interesting.
Nice.
I also like this morphology where it's got like this sort of dual concave forms. So I'm assuming the current comes right across there.
Yeah.
And it catches it from both sides.
Do you think we could do a little kind of fly over the top of it?
Sure.
I'm just thinking that we're doing such a beautiful job of getting video around it that could be a nice 3D model as well. Just get a little flash view of the top of it, then we kind of complete the coverage.
We have a question from the chat. They're asking if the coral reproduce. Does the coral reproduce?
How does it reproduce?
Or, yeah, they're asking if it reproduces, and if it does, how does it, I guess.
So we don't know a lot about reproduction in the deep sea just because it's hard to study. But oh, there's a fish over there, Jake.
There's
Yeah.
a fish.
But these corals can be brooders or spawners, meaning they can keep their zooids like in their tissues and release the larvae out into the world when they're ready, or they can just release their
Push in there.
zooids out and then have them mix in the water column. We know that a lot of shallow water species do this on a lunar cycle, but that doesn't always make sense for animals down deep. And it's really hard to know when these animals are actually doing that. I know there's a couple scientists trying to understand sclerotinian reproduction, and they might be very interested in collecting a piece of this for that research.
Thank you.
Oh, there's a little sea pen too.
Yeah.
I believe that if we did collect it, they wanted that specimen to be preserved in formalin to preserve the eggs or genetic
Okay, come on.
things. Not genetic things, like the structures.
Hang on
But
one
I don't know
moment.
much about that. It was just in the list.
We might, if it's not too late, Jeff is interested in a possible collection of that fish.
Yeah, we are stopped here.
Okay, great. If we could try to locate that one again and see what we can do. All slurp chambers are open and fair game.
Roger that.
All right, I've got slurp jar number 1 ready to go.
Roger that.
Would you like the pressure supply up a little bit
Yeah.
for this one? Okay, I'll crank
Pump
it.
it up. More beans.
Excellent.
More bean.
Done.
This is going to be interesting with the 1,000-year coral to the left.
It's going to be great. It's going to be good. All right, I am turning on suction.
No.
Yeah, this is Cuskill. Yeah.
Not gonna be able to reach from here. But can see the arm up against the,
Oh, yeah.
and then the left side is the big coral. So.
All right, so I think it's heading too much towards this coral, and we don't want to damage it. So I think we can probably skip this. Jeff is saying it's probably not worth it.
We might see another one.
Yeah, if it were to kind of move out and get in a better spot for collection, that would be great. But
Okay.
that's fine. Thank you. You can just X out of the sample. Perfect. Yep. We should see several more as we come up slope.
It'd be really cool if we saw like big reefs of that coral all over the place.
That would be really cool. Yeah, I'm really interested in how the currents are moving through this really beautiful, complex carbonate terrain. Seems to be a lot of organisms that found their special current spot. As we come up to waypoint 3 pretty soon, then the slope of this seamount is going to get a bit more steep, and we'll be kind of heading up that steeper slope until we get into the shallower depths of around 700 meters, 690 meters at the top.
Wow, there's a lot of brittle stars on that Walteria.
Want to push in there?
Yeah. This looks kind of like what we saw at the beginning
Yeah.
of our watch, doesn't it?
Yeah, this is the same type of Walteria flemingi. And it's got lots and lots of brittle stars on it.
Coming down a little bit.
Oh, there's a shrimp in there. So many amphipods. Like, this is the place to be. It's a party.
So they are coexisting, right?
Mm-hmm.
Yeah. Yeah, all these organisms that are living on or in another larger organism we refer to as associates. As they're associated with the sponge or the coral or whatever organism that they're living with.
Already?
Yep.
Come back wide.
And previously in the dive, we collected some really nice coral samples, and several of them had associates. We had an entire yellow coral with an anemone and brittle star associate. We got a piece of a bushy Stauropathes coral, some zoanthids with really interesting polyps, a Europticus black coral, part of a whip coral, and possibly an Acanthogorgia, and I think a Plumerella as well. So a lot of these coral samples, and as well as our rock sample, were collected in collaboration with our scientist here in the control van and our scientists on the ship, but also all of our scientists ashore. So we have a whole group of scientists from around the world, from different institutions, who are part of this expedition as well, and who are on the line with us at all hours of the day when we're diving, giving their expert advice and identifications and requests for samples. So the ship can only carry so many people, can carry just over 50 folks on board, but we're able to have many, many more people involved with what we're doing in real time, live. So just a shout out to a few of those scientists ashore who are currently with us right now. We have Jonathan Womack, who is an independent researcher, Les Watling from the University of Hawaii at Manoa, and also Piera, who was on board on the previous expedition at Pickrick. So she's tuning in as well with us. And earlier in this dive, we had Steve Oskovich at the Smithsonian Institution, and Chris Kelly as well, and Asako Matsumoto from the Chiba Institute of Technology has been tuning in quite regularly
Can
as
we
well.
look at the yellow one?
Yeah, let's take a zoom. So thank you to
Yes, thank you to all the scientists
all of our
tuning
wonderful
in.
folks, experts from all over. We really appreciate your input. Oh, who was that?
Push in there.
There's a little carrier crab.
Oh,
It's an enemy.
oh my gosh, my
Oh.
favorite.
What?
It's on the move. It
Ooh,
says
the anemone.
Lamoha.
The anemone looked like it was kind of hanging by a thread.
Oh, they kind of wear it like a hand muff kind of situation.
I really like those carrier crabs. So, as the name suggests, these crabs like to carry an anemone or a coral piece or something like that on top of them, and we've seen quite a few in the first Lebu's Voyage 1.
So this is a coral in the family Acanthogorgiidae. It is, well, we formerly called it Plaxaurid, but everything's been rearranged a little bit. But it is not Acanthogorgia. It is a different genus that is currently unknown to me. I think we collected some of these on the previous cruise, so hopefully we'll get some scientists ashore to be able to look at these specimens and provide some insight into the genus and species of this one.
Okay. Come back wide and pick up, keep moving.
Nice. That little carrier crab that went running by, Jeff thinks it might be a homolidae.
Yeah. So it's a crab in the family Homolidae called Lamoha.
Ooh, looks like we're gonna come up upon quite a tall.
Oh yeah, there's a
pink.
Paragorgia up there.
Yeah. Look at that. There's its base. Nice.
And then we can see some snake stars in the branches of this Paragorgia bubblegum coral.
Another food reference.
Yep. We do like our food. Well, I mean, to be fair, it is pink like bubblegum, and it has these sort of round balls at the ends of the branches that kind of
Want to push in?
reminiscent of bubblegum.
See what Megan's talking about.
It's easier to see when the polyps are closed, but it's really nice to see these open polyps. You can see each one of those little tentacles. You can count them. There are 8 per polyp. That's how you know that this coral is an octocoral.
All right, come on.
Did you want me to come wide?
Yeah,
Sorry.
you can come wide, Aaron.
Okay.
Thanks.
So we have a viewer that's asking, are we getting into the bacterial mat territory? Is that...
Ooh, it's a great question. So what we're looking at here is mostly sediment-dusted carbonate seafloor, and there is microbial life living throughout the ocean in all different kinds of environments. So there certainly are microbes and probably some bacteria living in the sediment here, but I think maybe what that viewer is asking more about is bacterial mats that we might see at hydrothermally active areas, which we won't be seeing on this dive or in this region. But on some of our previous expeditions and some of our highlight reels and videos from hydrothermally active areas, you can see some really exuberant microbial mats of all different kinds of textures, which is really neat.
I see.
Oh, let's look at that pink one.
Yeah,
That's different.
that does look quite different.
I think it's a Swiftia. So this is a coral. I want to look at the polyps a little bit.
All right, you can push in there.
Yeah, so Swiftia is a coral in the family Plexauridae, and it's very distinctive in that in most occasions on this coral, the branches come out at 90-degree angles from one each other, or more or less, and the polyps tend to be more of a yellow or orange tint compared to a very pink or red vibrant branching. So this is one of the corals that are still in the Plexauridae, and haven't moved out into a different group. And I'm also seeing a number of amphipods and I think a snake star. That's good, thank you.
Okay, come back on.
Oh, maybe it's not a snake star. Maybe it's one of those mollusks that are they don't have shells. They often feed on corals. So aplacophorans are in the mollusca, but they are without a shell.
Seems like a nice little perch up here.
Yeah, they look like little stools.
Yeah. Pillars.
Kind of reminiscent of some of the features we've been seeing above water.
Yeah, it's true.
There's some dead coral on that madrepora. So this was likely a large colony that broke apart. The coral itself is very fragile. It breaks very easily because it's made out of carbonate.
We're just arriving at waypoint 3, and this next section is going to get a little bit steeper. So we'll see how the terrain changes and if we get any different kinds of abundance of species or features that are jutting out into the currents as we move upslope. Looks like another couple of corals were coming up here.
Mm-hmm.
Nice. Oh. Oh. The purple one, past it. No, it was a little bit lower. Bamboo coral, anemone.
I'm going to come down a little bit.
Okay.
It's like everybody found a home in a pocket somewhere.
Yeah.
Different types.
Yeah, so a lot of the life that we're seeing down here, all of these sessile organisms, they're looking for food and something to grow on. So this hard substrate, this rocky bottom, is really ideal for them to grow on. And if you combine that with position in currents where there's nutrients and their food, that's going to be ideal conditions for all these kinds of corals and sponges. So wherever their position kind of gives us a clue in terms of the direction that their fan is oriented at, or...
Push on in there, Aaron.
Wow.
Wow.
This is one of the samples that we took in the previous...
Yeah.
I saw it hanging in the wet lab.
Exactly.
Uh-huh.
Yeah, it's really beautiful.
With the purple. Wow.
There's a few things happening here.
Definitely.
Yeah, so what's going on is there is this dead sponge stalk that used to be a Tretopleura, and it's been colonized by a number of different organisms, including this purple stoloniferous octocoral Clapularia, as well as this Pereid glass sponge that we saw in super high abundances on the previous cruise. Well as some other octocorals, some amphipods, hydroids, and foraminiferans. So even after its death, this sponge is providing a lot of habitat for a number of different species.
All right, you can come out wide.
Yeah, so a lot of these species are latching onto carbonate that's in a good position to the current, but if there's
So we
a
can
old
catch.
dead stalk of another organism that's already done part of the work, getting into a good current flow, then growing off of that positions that sponge in a even maybe more opportunistic area, and that's a crunchy one.
Yeah, it is a crunchy one.
If we had to put it in our crunchy and squishy categories, it's definitely a crunchy one.
Can we look at that white coral on the left?
Yeah, let's do it.
I think that is a sclerotinian coral called Enelopsamia.
All right, you can push in there.
Yeah, so what I'm looking at is the polyps. You see that they have more than 8 tentacles. They're not pinnate. So that's telling me that this is actually a sclerotinian coral, not an octocoral. That has a hard aragonite skeleton. And it's one of those sort of reef builders of the deep. Some dives, we see these in super high abundances. All right, thank you.
Okay, come on.
These crinoids are cool.
Yeah, they are.
I believe they're in the family Thalassometridae.
Is that the fern-looking like ones?
Yeah, those like very feathery feather stars.
So we have a question about anemone and if they move or they just grow where their larva lands.
A lot of anemones can move, and they will release their base, and some of them can somewhat swim to new locations.
Wow.
I've seen them do that. But most often they'll stay put where they are. It's a lot of energy to move somewhere else, and there's no guarantee you'll land somewhere better.
Thank you. Is this a crack?
Yeah.
Yeah, this is a fissure running through this area.
It looks like a little river of sediment. Let's look at the star.
Okay. Star it is. All right, push in there.
Like holding onto something?
Oh, it's eating a coral.
Oh,
That's what it's
eating
doing.
it. Ah.
Yeah, so these cookie stars, the Gonia asterid sea stars, are coral predators, and this star is definitely feeding on that coral that you see right there behind it. What they'll do is extend their stomachs out of their bodies and around the thing that they wish to consume and slowly digest it.
Wow.
All right, thank you.
Okay.
Oh, shrimp. Ooh.
This is a Paracleptrophora, a type of primnoid coral. They can grow in very, very large fans. You often see them on the tops of seamounts.
All right, I'm gonna come up a bit. Get back out in front.
Fish.
Fish. Oh yeah, there's a rat-tail fish, grenadier.
Nice. We can get a zoom if possible.
Go for a zoom there.
Oh, it's got a really cute, huge eye.
Oh yeah. Nice. Jeff will like that.
These fish are very, very fast and agile swimmers.
Oh.
Really nice zoom.
Hello.
Did Jeff say that this is a Kumba?
He's typing. Yep, he's a Kumba. He wishes we could collect it, but I think he probably
Yeah,
knows that it's too
it's
fast.
too fast. We've chased them before. They're
Yeah,
just
yeah. We
they
know
know
the
what's
game.
up.
This terrain is really ideal for fish hiding, and all those fast fish are not going to be slurped in time.
No, they can go straight into the little crevice. No, they know where to hide.
Yeah. That's how getting really nice video is really useful, as well as eDNA, which we don't actually need to collect an organism, but can sort of get a signature for that general biodiversity.
Ooh, yellow.
Oh.
That's a bamboo.
Yeah, so I was just chatting back home with our lead scientist, Igalo,
Nice.
and we had already decided that in addition
That's a big
to
one.
the predetermined interval sampling, if we see, oh, another fish, or the same one?
Yeah, it's the same kind as the one we were just looking at.
Nice.
Yeah, so if we see a bunch of fish, so the eDNA that primers we're using are designed to pick up vertebrates. So even though we saw the really beautiful coral earlier, the type of eDNA we're collecting would not be best for that.
Okay, that's good to know. So is there a certain kind of abundance of fish, or if we just passed a couple of fish, like even like right now, would that be kind of an interesting zone for Ecolau?
I think maybe in your discretion, if passing 3 fish is a lot of fish in one area, but you know, as
Sure.
for the deep sea, we have a different idea of what biodiversity is, but that would be the main idea.
Okay, great.
Yeah.
Yeah. Well, let's hold off then, because I think as we come up slope, get into shallower waters, we might see more fish. But yeah, that sounds great.
Yeah, exactly. Something on the wall there.
Very cool. Yeah. So we have these predetermined depths, and we have a certain number of bottles on the vehicle.
Mm-hmm.
So Elsa was just sharing about, we have this extra bottle, so we get to make this decision of what extra Niskin we want to get in addition to all these predetermined depths.
And then I was reading about taking different rock samples. What were the different areas that you guys wanted to try and get samples from?
For the rocks, yeah. So basically, the interest in collecting rocks is mostly in being able to age date these volcanic features. So we like to collect a. It's kind of difficult to say fresh because everything around here is quite encrusted and old and weathered. But some sort of rock that is ideally relatively in place, which means that we know that it didn't tumble down the slope and wind up in a different area, but that it kind of came from the spot that we're looking at. So good candidates for rocks often are still partly attached, but maybe have a crack or something so that we're actually able to break it off. And when we take a rock like that, that basalt rock, we can then at a later time, not on the ship, will be able to be age dated and give us a sense for how old these different seamounts and ridges and features that we're diving on actually are. And that helps us also give a sense of the geologic setting for the biodiverse habitats that are within these areas. So when we take rock samples, ideally we don't need to take several at one depth or in one area, as we kind of want to characterize ages for different parts of the seamount or feature we're diving on. So there can be different lava flows, and they can be overlaying each other, coming from different time periods, and
Like one happened and then it
Yeah,
cooled down and then another one
exactly.
happened.
Yeah, so volcanoes can remain active for quite a long time. So if we're at the very bottom of a slope, um, or even much deeper than where we started on this dive, which was still relatively shallow for our depth capabilities. We can dive down to 3,000 to 4,000 meters, and we started this dive at just under 1,600 meters. But yeah, we're just looking out for good candidates that are 1,000 meters or so apart, or several hundred meters apart. But in this area, there's not really rocks around here that are
Easy to take off.
yeah, they're not really looking like good candidates to me. And I was just kind of looking back in our log from some previous watches, and they did collect a basalt rock already. And that was just like 200 meters below us, so I think that's a pretty good collection that we have so far. On previous dives during the first Lebu's Voyage 1, during our first expedition in the Palau National Marine Sanctuary,
Oh, there's a star coming up.
we collected some rocks, and several of them were carbonates, so those aren't really useful for age dating. We're more interested in the basalt rocks.
Speaking of depth, we are currently at 1,098 meters down and slowly making our way up. We have about 500 meters to go till we reach approximately the
Push in
top
there.
of Seamount.
Yeah, exactly.
This is a slime star called Pteraster.
Nice. Slime star.
Is it slimy?
Yes, yes it is. If we collected it, it would just make a whole bunch of slime. Like buckets and buckets of it.
Huh.
Which is why we will leave it here.
Good to know. Good to know.
We don't want to slime up the lab.
All right.
So we have like crunchy, not crunchy, slimy.
Yeah. Texture is important.
Yeah.
Well, that's an interesting star. Let's zoom that real quick.
Bounce. All right, push in there.
This is a cookie star called Bathyceramaster.
Oh.
Great, thank you.
Okay. Back wide.
So here we have another Paracleptrophora with a Sternostylus swat lobster and a whole bunch of ophiuroids, a Crotella feather star, and some yellow zoanthids colonizing on this coral.
Nice.
Here we're also looking at a stalked crinoid or sea lily. I'm really liking this dive.
Yeah.
It's got good pacing.
Good.
Yeah. Making some headway. I'm really interested to see what we're gonna see at the summit.
Yeah, me too.
Yeah. So for any of our viewers out there who are just joining us, or for those who are still have been with us for a few hours, we're heading up a seamount at the moment. We're around 1,080 meters deep, and we're moving up along kind of a sort of ridge-like feature up to the summit of the seamount, which we determined to be the most interesting path up this very freshly mapped feature that we just finished mapping this morning. We're within Palau's waters, just outside of the Palauan National Marine Sanctuary. We're keeping our eyes out for all sorts of corals and sponges and anemones and other kinds of life within this terrain, in order to get a sense of the abundance and biodiversity within these.
There's a fish.
Within these waters. We're also particularly looking out for fish.
Push in there.
Yeah, let's take a good zoom. It's a rat tail.
Yeah, it's another one of those Kumba.
Kumba. What do you think about collecting this?
Oh, try for a suction?
Yeah.
We could try, yeah. Come wide on the...
Are we moving right now, or could we?
I can stop the ship.
Yeah. Why don't we try for a collection?
Bridge now.
Yeah, looks like a big one. But
If we could start with just a zoom on the
Try
head.
hold position.
okay, push in there.
Thank you.
Okay.
It's quite big, yeah. It might be too large for slurp, but
Yeah, well, the bigger they are, the better swimmers they are.
yeah. Also,
Yeah.
if we don't collect it, let's just try to get a nice zoom.
Still have the net.
If it swims away, then
Yeah.
no worries. That's already really great.
Oh, wow.
Some really big eyes.
Yeah.
Yeah.
I just really like their little barbels on their chins.
Yeah, looks
Come
like
on.
whiskers.
Yeah, I think that's going to be a bit too big for the slurp.
Yeah, agreed. Could we get a zoom on the belly by chance?
Belly.
Yeah,
Try.
if it might be too tricky or too late, but
Need some cooperation from
yeah.
the subject. But
Yeah,
Up higher.
Jeff is interested in getting some nice imagery of this. So one of our lead scientists, Jeff, is chiming in from another part of the ship. So Jeff's kind of trying to see
push in there.
the light organ, but these fish never seem to cooperate.
Oh,
They're quite swift.
so close.
It's all right if,
All right.
yeah,
Yep.
no worries.
Come on.
Yeah, thanks. A lot of other nice.
Oh, maybe.
Yeah. Oh, it's coming. Maybe. Oh.
Yes.
Close one.
All right.
Well, that was really great. Thank you, pilots and video, for
Good shot.
a lot of these really swift swimmers and species that are either too fast or too big to collect. It's really helpful to get great imagery and video to get documentation of the morphology and get a better ID on what we're looking at. So if you're joining us from anywhere in the world, feel free to ask any questions. We're happy to talk about what we're seeing and what we're hoping to see. We have a whole group of different folks from different backgrounds and expertise here, and as we're moving upslope, we're just keeping our eyes peeled for interesting things to zoom on, new species or organisms that we haven't seen in this area, or organisms that we've seen a lot of in this area.